In humans, the hemoglobin protein in red blood cells, which transports oxygen, includes two beta hemoglobin chains. Beta hemoglobin is a protein, and like all proteins, it consists of a specific order of amino acids. Now, humans are not the only organisms to possess beta hemoglobin. And when one considers chimpanzees, they possess the exact same amino acid sequence in their beta hemoglobin gene. Gorillas are also highly similar to that of humans and chimps, but there is one position where there is a difference, where the amino acid found in gorillas is different from that found in humans and chimps. Orangutans possess beta hemoglobin, but there are two differences from the human chain. Gibbons possess three differences from the human beta hemoglobin chain. An old world monkey might possess 7, a new world monkey might possess 8, a lemur 25, a mouse 28, etc. Now, it is possible that organisms would possess completely different proteins to transport oxygen, especially if they are unrelated. There are different molecules which can transport oxygen. It is also possible that organisms which possess beta hemoglobin to transport oxygen would all possess the exact same amino acid sequence if the protein was doing about the same function in each of these organisms. What is observed, however, is that the relationship between these organisms determines how similar their gene sequences are. Over vast periods of time, random mutations have put in certain amino acids in one position or in another, and that the longer it has been since two organisms shared a common ancestor, the more likely their amino acid sequences are to have accumulated differences. And so therefore, the comparison of these sequences suggests the relationships which are comparable to those obtained when comparing anatomical features or when studying the fossil history. Although some have used this to construct molecular clocks, it is difficult to try to pin down the exact timing of events simply based on the idea that amino acid substitutions may occur with such and such a frequency per million years, simply because any selection which occurs on these molecules can change the rate of substitution. So attempting to trace relationships or to form a molecular clock only works when the molecules in question have undergone equivalent amounts of selection and they serve the same function in different organisms.